Glucose can be broken down in the cytosol to produce a small amount of ATP. This initial stage of glucose metabolism is called glycolysis, which means sweet, glyco, splitting, lysis. Enzymes in the cytosol convert a single glucose molecule into two smaller pyruvate molecules. The energy released by breaking bonds is used to make ATP. Glucose is a 6-carbon sugar with a ring-type shape. The first glycolytic enzyme that it interacts with is called hexokinase. The prefix hexa, or hex, is derived from the Greek for the number 6. Notice that our 6-carbon glucose molecule is also represented by a hexagonal ring structure. Thus the name hexokinase refers to the glucose molecule that it interacts with. Hexokinase phosphorylates glucose to produce glucose 6-phosphate. The phosphate group is derived from cytosolic ATP, producing ADP and consuming energy. A second cytosolic enzyme alters the structure of glucose 6-phosphate to create an isomer with a pentagonal structure, fructose 6-phosphate. Subsequently, phosphofructokinase phosphorylates fructose 6-phosphate to produce fructose 1,6-bisphosphate. Again, the name of the kinase refers to the molecule that it interacts with, a phosphorylated fructose molecule. In a series of additional steps, glucose is broken down further into two 3-carbon molecules. 2 molecules of NAD are reduced to NADH, and 4 molecules of ATP are produced. At the end of glycolysis, we are left with 2 pyruvate molecules. If the cell is undergoing aerobic respiration, the pyruvate molecules will be transported to the mitochondria, where they will be involved in the production of much more ATP. In the absence of oxygen, the cell may undergo fermentation. The rate of glycolysis is controlled to meet the energy needs of the cell. Glycolytic enzymes are inhibited or activated at allosteric binding sites. The most important rate-limiting enzyme is phosphofructokinase. When cytosolic levels of ATP are high, phosphofructokinase is allosterically inhibited by ATP. When cytosolic levels of AMP are high, phosphofructokinase is allosterically activated by AMP. Thus, phosphofructokinase is very sensitive to the ratio of ATP to AMP in the cell and can be turned on or off to regulate the rate of glycolysis when the cell's energy needs are high or low. Hexokinase has an allosteric site which binds glucose 6-phosphate, its product. Both the conversion of glucose to glucose 6-phosphate by hexokinase and the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase release a large amount of energy, making these reactions practically irreversible. The product, fructose 1,6-bisphosphate, is only used in glycolysis, making it especially important to regulate the glycolytic pathway prior to its production.